For those of you who aren't familiar with CF, it's the most common serious inherited disorder, particularly prevalent amongst Caucasians. In the UK, where I'm from, it's about 1 in 20 individuals are carriers. There's maybe 50, 60 people in the room, which means there's about 3 carriers in the room. You find each other in the bar tonight, and you have kids, then there's a 1 in 4 chance those kids are going to have CF. Your chances are slightly better than that, because I know I'm one of the carriers. Carriers are generally unaffected by the disease and tall and handsome and witty. Worldwide, there's around 1 in 100,000. There's about 100,000 individuals affected by the disorder. There are different ways of thinking about the impact on the lives that the disease has. The median survival of a child born today is about 40 years, and that sounds pretty good. It doesn't sound such a terrifying disorder. However, actually, the age of the people dying in our hospital is more like 28, 29 in the UK. And one of those dies every week or so, and that's a pretty miserable time, a period of your life to be dying. I remember being mid-20s was really quite a fun part of my life, and we need to do more. It's a disease of epithelia. There are many organs affected, but the biggest problem is that lungs accumulate sticky mucus, and that sticky mucus encourages bacterial growth, and repeated in bacterial infections leads to lung destruction. And our patients die basically being unable to breathe, not getting enough oxygen into their system. The gene was cloned back in 1989. It's got a long fancy name, which I'm not going to say. I'm just going to say CFDR for the rest of the talk. And CFDR is an epithelial chloride and bicarbonate channel, and that's its job. I'll tell you a little bit more about its biochemistry. CFDR's job is to maintain the height and the composition of the fluid in the airway surface. So here's a diagram of the conducting airways. It's the conducting airways, the main tree-like parts of the lung, which are a problem in CF. The little leaves where all the oxygen gets exchanged, they're fine in CF. Here's a cross-section of a respiratory epithelium. And on the surface of respiratory epithelium, there's a tiny fluid layer, airways and surface liquid. And CFDR sits... I was told I was going to have a magic pointer, but I don't consider it'll make that work. But okay, whatever. CFDR sits. I can't see a thing from over here. Oh yeah, there we go. Here we go. So CFDR is located on the apical surface of these cells and sits around about there. And why is that important? What does it do? Well, these are cultures of airway epithelial cells that you're looking down on top of. And on the left-hand side, we have cultures from a non-CF individual. On the right-hand side, we have cultures from a CF individual. And the cultures have been sprinkled with fluorescent beads. And in a circular dish, you can see that that cilia beating moves mucus and moves things that we breathe into our lungs consistently out of our lungs, and that's a really good thing. If you have CF, there's not enough fluid on the surface of those cells. Those cilia collapse, and there's not mucociliary clearance. And the bugs bugs and everything else we breathe in, all the other gunk that we breathe in, is not effectively removed from people's lungs. That's the kind of fundamental issue. Now, a beautiful experiment done by Larry Johnson many, many years ago showed that if you mixed in between 5 to 10% of cells which express CFDR into one of those sorts of epithelia, you can effectively correct the entire epithelia. So that's our therapeutic target. You need between about 5 to 10% of the cells to express CFDR to get back, in this graph, ion channel activity, but in other measures, the similar sort of percentage of cells seems to be important. The very first experiment I did in a CF mouse about 1,000 years ago was to take trivial CFDR expression plasmid, mix it with commercial liposome, introduce it into the lungs of that mouse, and astonishingly, that actually largely corrected the ion channel defects associated with CF. And tragically, I've spent the last 20 or 30 years trying to translate that relatively straightforward experiment in mouse to try and do that same thing in people. So unfortunately, the rest of this talk, you may want to sit there and say, he's particularly hopeless at doing that. He's taken so long, and he still hasn't really got to the end, if I don't want to spoil the punchline for you. So what are some of the challenges? And that was one of the things that the chair asked me to address during this presentation. So the challenges to doing these kind of things with non-viral systems. Well, it starts out in CF with the biology. And the biology of the airway epithelium is that it's slowly replaced, and there is no key underlying stem cell. We don't have one target cell we could take out, we could treat, we could put back. Instead, There are many, many different cell types that make up the lung. There are some common ones, there are some rarer ones. There are multiple different surfaces. There's the air surface, the blood surface, and so on. And probably all of those geographic places in the lung have a particular cell type which is capable of expansion and replacing the cells. But there isn't one particular cell type. Many cell types have that ability. Overall, the half-life of the cells in the lungs, there's some arguments about that, but it seems to be of the order of a year and a half, something like that, in an unaffected individual. It's probably less than that in an individual with CF. But nevertheless, if you're thinking about doing a gene transfer experiment, or even a gene editing experiment, you're going to have to go in and replace those cells because there is no underlying stem cell that you can treat and fix. So you're going to need to have repeated administration. That's going to be a key point I'm going to come back to throughout the talk. The clinical experience was there was a bonanza, a huge rush into CF gene therapy. It was such an obvious, trivial target, put stuff back into the lungs. As soon after the gene was identified, multiple groups tried multiple different gene transfer vector systems, particularly in the US, adenoviral vectors and AAV vectors were tried, particularly in Europe. I don't really know why the distinction, but particularly in Europe there was a focus on non-viral gene transfer. And there was a rush of clinical trials in the 90s, all of which frankly gave near enough the same result, which was there was evidence for modest gene transfer and there was transient correction of that CF ion channel defect that I was describing. That's a tragically old photograph of me doing some of the measurements in one of these clinical studies. I noted in my wardrobe this morning I had the same blue shirt, so I put a different one on today. What this picture is highlighting is one of the key measures in these clinical studies is a test called nasal potential difference. It's not a very elegant thing to do. You shove a probe up someone's nose, you put them in front of a high-tech washing up bowl, you flood the surface with liquid, and you can measure how much electricity, which is a byproduct of how much chloride is being moved across that surface. And if you have CF, then you have very little electricity in your nose, and if you don't have CF, you have more electricity in your nose. It's a very straightforward distinction between those. And without any reservation, the very best result I've ever seen in a CF gene therapy trial in that nasal PD experiment is shown on this slide right now. Complete correction of that CF defect, having put plasmid back in expressing CFDR. So, fantastic. However, that effect in that particular study was pretty transient. It only lasted about a week or so. And again, that goes to another issue that the chair asked me to address, which is about duration of expression with non-viral vector systems. The other issue that we found when we tried to do this experiment in the lung rather than the nose is we get the same sort of effect. You can see correction. But what I should have said was that was the very best experiment that I ever did, and it doesn't happen all the time. That happens maybe about a third of the subjects or so, and there's much less pronounced effect in the other two-thirds of individuals. And that variation is an important thing that we need to understand too. But the other really different thing, the really important thing that happens when you try and do that experiment in the lung is that you see inflammatory responses, flu-like symptoms as a result of delivery of those kind of vectors. Now, some time ago, the CF charity in the UK came to the three groups that had done effectively the same sort of experiment and said, for goodness sake, guys, can you stop repeating each other's work and try and work together? And once we got our egos out of the way, which did take quite a while, we formed a single consortium where the three research groups in the UK who had all done separate clinical trials came together, and we have a single translational program. It's the groups in Edinburgh run by Chris Boyd and Jerry McLachlan, in London run by Eric Holton, Jane Davis, Nita Griesenbach, and in Oxford by myself and Deborah Gill. And we gave ourselves the relatively straightforward task of identifying the very best possible gene transfer systems for the lung. And they had to make sense in terms of in vivo studies that worked in mouse and in sheep. It had to work in a pharmaceutical development suitable fashion. The drugs had to work on repeated administration. You had to be able to manufacture them. They had to be stable in the pharmacy, those kind of things. And the obviously target product profilers need to express at least 5% of the cells need to be expressing CFDR. And ideally, we like to deliver that using an aerosol, which is a really simple way of delivering our stuff into the lungs. Now, we focused on non-viral gene transfer elements for reasons which are reasonably obvious to most people in this room, but it is basically if you use viral gene transfer vectors, they just don't work when you give them lots of times in vivo. Here's some experiments using AAV, but we've tried all sorts of other gene transfer vectors as well. Great gene transfer the first time, modest, some degrees of evidence of gene transfer a second time, no gene transfer at all by the time you're giving a third dose of these vector systems. And that's because we have an immune system and we generate antibodies against these kind of vectors. There is an exception to that, which I'll mention to the end of the talk. But largely, the vector systems that we analysed in those studies all did the same thing. And so our focus in terms of identifying a really good non-viral system was to use a screen of a large variety of commercial and academic non-viral gene transfer vectors. We tested them in mice, we tested them in sheep, and we were looking, as we were saying, to make sure that basically that they worked. There was the list of the gene transfer vectors that we started out with. We did an in vivo mouse lung screen using all of those, and what we found was that, you know what, in vivo is completely different from in vitro. Forget doing anything in vitro is a complete waste of time. We found that instilling these things, using a bolus of fluid into the lungs of mice, is very different from administering these things with aerosols, particularly with respect to the inflammatory responses. And aerosols are much more benign, and data that you get by putting a bolus of liquid into data can be quite frightening, but actually data you generate with an aerosol, oftentimes gene transfer agents are much less inflammatory. Of those three, sorry, I should have said that there were three winners. Oh, I've got the wrong button here. There were three winners that came out of that list: a particular version of PEI, a liposome formulation from Genzyme called GL67, and a reagent from a company called Copernicus at the time called Plasmin. When we took those gene transfer agents into the sheep, which was our large animal model of choice, this GL67A formulation was a clear winner from those experiments. And so we've gone on and focused our research on this formulation of GL67A. So, oh my, what beautiful, in UK ticks, in US check marks have been turned into wicked boxes, but basically it ticked all of the boxes necessary that I've been outlining in terms of our drug development. And the three things that I'm just going to focus on is in terms of dealing with it in terms of pharmacy, dealing with those in terms of gene expression, and also reduction and elimination of flu-like symptoms using this GL67A formulation. So what is GL67A? It's a mixture of three molecules. The key one is a cholesterol derivative, and then it's also mixed with some of our friends, DOPE and DOPE-PEG, mixed up and freeze-dried out into single-use vials, which have to be resuspended close to the usage time. When you look at those liposomes by themselves, they generate beautiful, simple liposome structures. When you mix them with DNA, magic happens. I've never understood what the active components of these things are. It's somewhere in there. If answers on a postcard, please, if you can explain to me which is the important bit of these formulations. Which leads us to a really important finding, which is that when you try and do these kind of, use these kind of gene transfer reagents in a trivial tissue culture experiment, they're dead easy to prepare in a mix. You're mixing small volumes of these reagents together, they behave perfectly. As soon as you start trying to think about mixing large volumes of these reagents, they stop behaving properly. And our standard preclinical aerosol mix was 10 mils of these formulation, which included around, I can't actually read it and I don't remember, but a whole bunch of plasmid DNA and about 150 mgs of the combined lipid as well. And when you mix that, most of the time everything is fine, but some of the time it's really bad. And around 5% of the time when we were doing our first set of mixing studies, we'd see these ghastly complexes. No way am I going to put that into an animal. There's no way going anywhere near an individual as well. We had to get much more consistent and pound of preparation of these reagents, which required two things. One was an investigation of the contaminants within our plasmid DNA, and what we noted was when you start purifying DNA, actually the amount of salt in your preparations is alarmingly variable. And the way we addressed that was pay a lot more attention with our formulation effort, and rather than using water to finely formulate our DNA, using just a tiny bit of salt, 8 millimolar salt, actually massively reduces the variation and decreases the overall range of salt in that thing, and that made a big difference in terms of our complex formation. Another thing is we spent some time in the hardware store. If you try and mix two things together, you need to do it uniformly and consistently, and the hardware store guys know everything about this when they're using two-component glues. And the reason that two-component glues work really well is that you use things called static mixers, which are kind of like two spiral plastic components over which you can pass two solutions that will be homogeneously mixed at the end of the process. So we very much adopted using static mixers. The regulator said, how fast are you going to push those mixers? So we had to come back and design a piece of equipment that would push our device, and a disembodied hand will come in here and we'll take two liquids and turn them into one completely homogeneous thing. And in clinical use, we've made over 700 clinical lots, 0 failures, none of those. Whoa, sorry. None of those disasters which I showed you previously. And this kind of approach is terrific for mixing reagents close to bedside if you need to use them like that. So I can prepare my things under pharmacy conditions nicely. In terms of duration expression, this is all about plasmid DNA. The first plasmids that we used were completely riddled with CpGs, CG dinucleotides. And for those of you who aren't familiar with the biology, they're rare and methylated in you and I, and they're common and unmethylated in bugs. We manufacture our DNA in bugs. And they're recognised by host defence systems that say, help, help, I'm being infected by bacteria. And so we respond as if we're being infected by bacteria with inflammatory reactions. It's the right thing to do if you think you're being infected like that. But that's the driver of the flu-like symptoms in our early clinical trials. So, easy, you just take all the CG dinucleotides out, right? No problem. Actually, completely straightforward in terms of cDNAs, really difficult in terms of origins of replication, and really, really difficult in terms of promoters. But nevertheless, when you do that, by whichever metric you choose to use, you can abolish lung inflammation after you've administered those kind of reagents. And as a bonus, something that we didn't factor in at all when we started down that road, we also massively increased the duration of our transgene expression. So we got a 2-for-1 result on that. So in terms of applying these things in a clinical fashion, the question we wanted to ask was, does repeated application of these kind of reagents lead to a clinically measurable benefit in our CF patients? And obviously the way we need to do that is in a multi-dose clinical trial. We had to make sure that we understood what a single safe dose was, and so you have to do a single dose trial. And we had to understand what the optimal patients and the endpoints of doing that in. And so we ran what we called a run-in trial, where we took a whole bunch of CF patients, measured a huge basket of potential biomarkers, and worked out which ones we thought would be appropriate for application in those clinical studies. That longitudinal study, as I've outlined, used a wide range of efficacy assays that we thought. We had over 200 CF patients for 3 years, and the job was to identify those patients in which we were able to deliver our reagents, and yet we still had something to measure. Obviously, if you put into a pristine lung, there's no defect to measure, but you can probably get your reagents in more readily than if you put it into a terrible lung, where there's lots of things to measure, but you may be obstructed physically in terms of gene delivery. And what we found from that study was actually rather disappointing. What it said is the best way of measuring whether someone has CF is a really boring assay. It's called FEV1. It's how much air you can blow out in 1 second. The trouble with FEV1 is you need loads of people in a clinical study to power it effectively. We had lots of other fancy ideas that we thought would reduce that number of people, but they just didn't pan out. You can power a study for a 6% change in FEV1, which is clinically meaningful, as long as you have about 60 people or so on each side of that clinical experiment. We did find a basket of secondary outcomes which might be interesting and useful, but all of those require substantially more people in a clinical trial before you can get statistically meaningful results. And so don't be surprised when some of my data is underpowered later on, as I show you, because there's only so many people you can put into your clinical trials. The single dose study was relatively straightforward. I've jumped over a thing which said, we had to work out a way of delivering these things using aerosols, and we've picked a particular nebulizer. And I could give you a whole hour on why we picked a particular nebulizer, but we won't for today. And we started out in this single dose study with 35 CF subjects. They had this drug delivered to their nose and to their lung. And we measured a whole bunch of things. The thing I'm going to focus on is that nasal potential difference measurement that I alluded to earlier. Before the treatment, as we've showed, in CF there's very little electricity in someone's nose. The trace takes about 5 or 6 minutes to show, and that's kind of what I'm showing here. And that's the sort of trace that you get with someone who doesn't have cystic fibrosis, lots of electricity in their nose. 2 weeks after giving them our new plasmid mixed with Cystic 7A, we see a kind of partial correction of that CF defect. 4 weeks we're up to seeing complete restoration of that defect. That's sustained out to 6 weeks, and it starts to fall away after 9 weeks. So again, we have extended the duration of that correction, but we've not fixed it for forever using those molecular tinkerings. But nevertheless, we've identified a safe and effective dose. We focused on using a 5 ml aerosol formulation, which had about 13 mg of our plasmid and about 75 mg of our combined lipid. In that single dose study, again, around a third or so of the subjects had striking changes in potential difference measurements. There was also a significant change in a measurement called lung clearance index, which indicated that the small airways of those lungs were being improved. So that moved us on to doing, that's kind of, the single dose study was shown to be both safe and effective, which set us up for multi-dose study. I still believe that this is the world's largest in vivo gene therapy clinical study conducted to date. We had over 1,700 patient visits. We had 15 UK patient identification centres. There were 2 dosing sites. You either got dosed in London or in Edinburgh, depending if you lived near England or Scotland. We enrolled subjects from 12 years and up. In the UK, people who are over 12 can give informed legal consent. They had between 90% FEV1 and 50% predicted FEV1. That was that range we identified in that run-in study. They were randomised 1-to-1, so approximately half got placebo, approximately half got the active drug. And they were given 12 doses of that reagent on a monthly schedule. There was some leeway allowed if they didn't have to come on exactly a monthly. There was, little gaps. But to be enrolled in to be accepted into the statistical arm of the study, you had to have received at least 9 of those 12 doses. So here's the Consort diagram. You probably can't read it. I can't read it from up here, and I can't quite remember exactly how it works. We screened a whole bunch of people. I think it was something like 2,000 people. Can't read the numbers. Lots of people got screened. We ended up with an intention to treat group of around 135 individuals, and 114 individuals met the statistical acceptance of receiving at least 9 doses of the gene transfer reagent over the course of that study. No bad things happened in the trial. Well, that's not true. There were some things that happened, but there were things like someone broke their leg going skiing. There were no drug-related adverse event incidents. There were some adverse events like that. The demographics of the study were particularly well matched. There are lots of numbers, I can't read them, hopefully you can, but what I can remember is that the groupings was actually done pretty well by that randomisation, and there were no safety issues addressed. So here's the killer graph, the FEV1 graph against time in this study. So up is good, bad is down. So if you take the people who got the placebo dose, which was a saline dose from that study, basically their lung function declined gradually over time. That's completely consistent with registry data. That's what happens to our CF patients. Their lung function gets worse. When you add the drug, that decline in lung function stopped. And that was the first time there's any clinical benefit been observed in a CF gene therapy clinical trial. Now, is this an astonishing drug? No. Is this a start? Yes. What we really want is we want to see patients gaining lung function, and that is possible. There are drugs which are suitable for a very small number of CF individuals, a small molecule which targets a small proportion of those, and they can see improvements in lung function. So clearly our gene transfer is not as good as it could possibly be. But nevertheless, it's a damn good start, I think. The secondary endpoints all followed the primary endpoints, so there's a good support for the treatment being effective and active. But as I outlined earlier, the majority of those particular secondary endpoint measurements were underpowered and therefore didn't reach statistical significance, though a few of them did. But what was nice is all of them went in the way of active. All of those measures of whether you were getting better was going in the direction of having received the treatment. That was actually really quite remarkable in terms of that sort of study. Interesting observation, if you're in the sicker half of our patients, those with less well lung function, actually you had a more marked change having received the drug, and there was some evidence for lung function improvement in those individuals. And we believe that's because actually the drug gets trapped in the right place, and therefore a higher effective dose has been delivered in those individuals. And that's something we'd like to look at in future studies. So in terms of thinking about what we've done here, we've identified a safe and effective non-viral formulation. It's suitable for all CF mutation types, which is not true of those small molecules that I was alluding to. It halts the decline of lung function. It's similar to current small molecules for common CF mutations. There's other drugs which are not quite as effective. And obviously the thing we'd like to do is increase dosing frequency and add small molecules in to see if we can improve the overall effectiveness of this class of drugs. So I've said we and I a lot, loads of people, as you might imagine, were important for doing that kind of study. All across the UK, from both, all 3 of our sites, Edinburgh, Imperial College in London and Oxford myself. I'd just like to acknowledge my research group, nice picture of them there, and our funders for these sort of studies, the Cystic Fibrosis Trust and the UK Department of Health and the National Institute for Health Research were particularly important for getting those studies done. I must acknowledge 2 people who were in the lab at the time but are now elsewhere. Ian Pringle, who was particularly important in terms of the molecular biology, and Lee Davis, who was fantastic in terms of the aerosol systems and many other things. Those two individuals also played a particularly important part in our viral gene transfer efforts, which if you want to hear more about, I know I shouldn't be using the V word in this meeting, but we have a bunch of posters and a talk from Joost on Thursday. Come by and say hello, and I'll stop there.
We have time for a couple of questions. So I'll start, Steve. I'll start. If you look at the patients that respond to the modulators, you mentioned that there's a small group of patients that are benefiting from these modulators now for the benefit of the audience. You can get this big bump in FEV1 early on, but it actually starts to taper off after a while. If you look at the studies that have been done with these modulators and you segregate the patients in those studies from milds to severes like you did with your study, are the responses in the right direction? Is what you've seen what you would expect if you have bad lung function and you get an improvement from there, whereas if you have a high lung function to begin with, maybe you don't get that much of an improvement?
I'm not sure I know the answer to that, but I can answer a different question, which is that in our trial, is our effect sustained? Mm-hmm. And it is remarkably sustained. So a bump in lung function can then be sustained for quite a long time. And so although there is a gradual decline, our patients 2 years after treatment, the active group was still clearly ahead of the placebo group. So it goes some way to suggesting that those sort of changes can be sustained. Stunned silence.
Thank you, Stephen. You have used one formulation made with a cationic liposome. Do you think it's possible to improve transfection efficiency of your formulation? And probably it's very difficult to start again these three. So
I have every faith that chemists can do more and wonderful things in terms of improving the ability of those sort of formulations to increase gene transfer. I think we're probably, personally, I'm probably done on the plasmids. That's actually my expertise if I have one. I don't think you can do a lot more. You can tinker around the edges, but I always think there's more that can be done in chemistry. And it's quite clear our viral formulations are capable of transducing more cells. So that means that the cells can be driven to take up genes under certain circumstances. So yes, I have every faith. You, the people, can fix this for me.
thank you.
Hi, Steve.
Hey, Steve.
Question about models. Do you think in retrospect, the mouse model was predictive of efficacy? You had some nice corrective data in CF mice. Do you think that really reflects the human lung, or are there other models?
The CF mouse is a really interesting one. You and I know there's a long history on those things, and in those experiments, later on, you come to realise that you aren't quite affecting the things the way you think you're affecting, and the impact of olfactory epithelium rather than the respiratory epithelium is particularly pronounced in those experiments. So there are some real questions about CF models. I think mice in general aren't people.
Thank you.
Honestly, if you went to every other talk in this meeting, you'll find that mice are equivalent to people, so I think it's an important thing to remember. And I think it's important to take your formulations for a ride in large animals. You've been famed for doing pig studies, and they're brilliant. We've often used sheep, and I don't think there's a lot of difference between those two, but I think a focus on large animals is a really important one.
Thanks.
Thank you, Steve. One question concerning the plasmid. Do you think that using a small one, a nanoplasmid, for example, what the one that James will talking about, it will be okay? And what about combining the lentiviral vectors as a first administration, and then maybe, I don't know.
Okay, so two questions there.
Yeah, there's two questions in 2 points in one question.
So on my other hat, I like lentiviral vectors, and I said that there is no virus you can repeatedly administer except lentivirus to the lung. And there's a whole talk I can give you about that as well. But a particular pseudotype of lentivirus has the ability to repeat administered to lung epithelium because probably it transduces cells incredibly fast and can beat the immune reaction.
I thought about do one lentiviral vectors and maybe boost with, maintain with a non-viral one.
Yeah, that's a really kind of really interesting. We often thought about introducing a receptor that you could then target, that kind of thing. You also talked about the size of plasmids, and I think there is stuff that could be done there, and I think removing antibiotic resistances is probably a good thing and so forth. In our hands, we haven't seen a pronounced effect of plasmid size on gene transfer efficacy with those formulations, but it may be that other formulations are more plasmid size sensitive.
Okay, thank you.
Thank you, Steve.